Hello students, let us go for understanding the process of maturation of mRNA or hnRNA. In the earlier diagrams we have studied there is DNA present, this DNA is found in nucleus. This DNA will undergo process of so called as transcription so as to form hnRNA. Now we will show in short the process of formation of hnRNA. So this is DNA present, DNA undergoes process of transcription. So by the process of transcription we can say what is formed? hnRNA is formed. So we will show presence of formation of hnRNA. It will be somewhat like this. We have to make a loop. Fine. This is DNA which is undergoing uncoiling process, and then this is loop, replication loop, and then okay. So here we'll get formation of mRNA from this. This mRNA is immature and hence is called as hnRNA, heteronuclear RNA. This heteronuclear RNA has got two regions present on it. There are certain parts which are not expected here, and there are some parts which are expected over here. So two regions are clearly seen. Some are called as exons and some others are called as introns. So this kind of parts we can label them as, these are exons, and there are some regions present, such kind of regions, they are called as introns. So process of maturation will include three stages. First, there will be removal of introns and retention of exons. Second process will be there called as capping, addition of MGTP, that is methyl guanosine triphosphate, and then third process will be tailing. Now this hnRNA, it has got two ends, that is very obvious. This is 5' end and this is 3' end. First process we'll call it as splicing. What is splicing? Splicing is actually process of removal of introns. So the entire structure will have such kind of structures, means the hnRNA will undergo coiling, and during this process of coiling the introns would come together like this, and there will be splicing of introns. So all these are introns. Again we will show these are introns. So introns will be removed, and then what remains is, again this is 5' end, this is 3' end. So result of this we can show is nothing but only exons are remaining. Now this is 5' end, this is 3' end. This entire process is called as splicing. Second process it follows is known as capping. Capping is an event where addition of MGTP will be done at 5' end. So this is, suppose, MGTP, methyl guanosine triphosphate. So after addition of methyl guanosine triphosphate, it will look somewhat like this. This will be 5' end, this is 3' end, and what is done? MGTP will be added here. This entire process is called as capping. Similarly, it will follow third procedure. This third procedure is called as tailing, where adenylate residue will be added. So here we'll show presence of this one, adenylate residue, and this adenylate residue will be added at 3' end. So then it will become like this. This is what hnRNA. Now we are adding this adenylate residue on 3' end. It will take place n number of times. So this is addition of adenylate residue at 3' end, and at 5' end already we have got a cap fixed. So after these three processes of capping, splicing, tailing, the RNA now formed will be called as mRNA, and this mRNA will be ready now for undergoing process of translation. तो अगर डायग्राम पूछा जाए, शॉर्ट में क्वेश्चन पूछा जाएगा व्हाट इज मैचुरेशन, तो उसमें ये डायग्राम सब ऐसे इस तरह से आप निकाल सकते हो. पहले थोड़ा सा दिखा दीजिए, ऐसे बुक में दिया हुआ है ये, ठीक है. सो डीएनए दिखाना है, ट्रांसक्रिप्शन लूप दिखाना है, उसमें से आउटपुट निकला, एचएनआरएनए दिखाना है. एचएनआरएनए के ऊपर दो पार्ट्स आपको मेंशन करने हैं, इंट्रॉन्स और एक्सॉन्स. इंट्रॉन्स को निकाला हुआ जाता है इस तरह से दैट यू कैन शो. सो इंट्रॉन्स आर रिमूव्ड, देन here you can show presence of capping. इंट्रॉन्स को निकालने का प्रोसेस स्प्लाइसिंग है. स्प्लाइसिंग के बाद ऐसे हो जाएगा, then capping and then tailing. So this is how you can represent the entire process of maturation of hnRNA. So the next session will go for making diagrams related to translation. Thank you.